Hi, I want to work on something with you that will allow you to better understand your reading from chapter 3, which is genetics, DNA molecule, and some of its structures, functions, and so forth. It also has an intro section on the cell in general and fields of genetics. Do remember that in university level classes, the instructor cannot go over everything in every chapter. So my lessons will focus on things that I have found over the years that students have a little bit more difficulty with, or that are structurally a little bit more complex. And I'm going to leave you be because I do trust that you can do your reading, that you can chart out or outline out your reading, and you know what to do with the rest of it. So this one is a activity type lesson, and I'm going to try to show you a way to chart out information on your embedded reading from about page 37 to about page 45. It's a lot of information, and some of the things that I do notice that frequently happen is that one, students just look for the definitions. That's not going to be enough for you to have strong scores in any of our type of activities for this type of course, because it's a transfer level life science course. Secondly, students do not look at the illustrations, the charts, or the tables that are embedded. And this way we're going to also emphasize how important those are, because in particularly these more compacted forms of reading, for introductory level courses, quite a bit of the key information is going to be there in a graphic rather than written in. And thirdly, this is one of those types of complicated core bases of learning where you start off with learning the names of, let's say, for instance, a structure, and it'll be quite a few pages later where they talk about what that structure does. So joining those together can be a challenge. So firstly, for you, best case for this type of learning is for you to get a longer rather than shorter piece of paper. If you have an 11 by 14 or a legal size piece of scratch paper, the back of something, perfect. If you don't, just open up a paper bag. It really doesn't matter. This is about physically using your hands, decision making, and coordination, color, all those sorts of things to put together something that's really going to help you to remember, understand, and remember. Why am I asking you to do it this way, by hand? One, not all of us have really fancy computer equipment. That might be part of it, but really, actually, empirically and scientifically, we do know that when humans use our hands to write, sketch, outline, recall, and that sort of thing, that the results empirically are stronger in assessments, in function, and later on in being able to remember it. So this is from everything from studying students that are all using a laptop to take notes, or many using a laptop to take notes as graduate students in some of the world's most elite universities in any language, all the way down to much, much younger students, and you would be in the group in the middle. So this is going to be an effective way to remember this in it, because I know a lot of you are going to end up taking more courses in life sciences. Okay, so firstly, take your piece of paper and fold it in half this way. And then, holding it this way, fold it in half one more time. You're going to have 4 long and narrow columns. Also for this one, best case scenario, be very careful with how tidy, neat, and visually clear it is. So for me, I'm going to use on the board behind me several different colors of markers so that you can see that things that match up on one side, match up on the next column, match up on the next column. If you're doing it all in just one color pen, it's going to work okay, but not as well as you do this. And secondly, as far as the structure of it, a straight line, which could just be another piece of paper, or it could be if you have a ruler or something else like that for it. Okay, so I'm going to create this basically back there for you. So let's go. All right, for very, very useful and helpful notes that are going to put together in charts, diagrams, and so forth, always label them. At the moment you're making them, it is clear that you remember what's going on. But once a day has gone by, we really start to lose information from our short-term memory. So unless we're really repeating it or working on it, then when we come back to it again, quite a bit is gone. And so firstly, this is the DNA molecule. So we are looking at the DNA molecule, deoxyribonucleic acid. We will compare it briefly on this chart to RNA or this lesson, but right now, DNA molecule. So the DNA molecule here is going to be something that we can take a look at with 4 columns. Right now, this first section, we aren't going to do a lot with the first column, but the first column I'm just going to say is other info, right? Like details. Next column, this is going to be vocabulary or terms. Middle is going to be the structure diagram. And the fourth one is going to be output. What does it do? So you're going to have those folded lines that you can use right here on this one. So an other information column, the vocabulary or terms, and then structure or output. Now, firstly, let's start one side of the DNA molecule here. You'll see in charts for courses like ours that we are not using the chemistry type of chart. So start off with this one, leave a couple up here that are really, really wide, and then as you get a little bit lower on your chart, closely fit them together. We're going to know that it's this way, but as you label it in the first time. So, a whole bunch of these little ones for this one. Okay, so the DNA molecule there. So those little lines versus tightly fit so that we can fold that in. Now, go take a look at your notes because you want to do this in concert with your reading. Imagine that. And DNA structure and function starts on 37 in your embedded course text here. And if you look down towards the bottom, there's figure 3.2, and it starts off saying DNA structure 1, the molecular level. So it says the structure of the DNA molecule is a particular vocabulary for shape. Well, I'm going to leave you to that one. That one's simple and you've already heard of it. And then it says the basic unit of DNA and RNA is a molecule called a nucleotide. So in your notes, that will be your first vocabulary term. So nucleotide. As you put something here, if it relates to this and this, keep it in the same color. So I'm starting off in blue, nucleotide. So on this chart, and I'm actually going to leave this empty here towards the tip and we'll start down here. A nucleotide, and you'll see it there in figure 3.2 and also in your reading, is composed of a sugar, which in DNA is deoxyribose, a phosphate, and something called a base. A sugar, a phosphate, and a base. Over here in your other info or details column, go ahead and be sure that you label over here and then finish this up on your own. DNA bases are, what are they? The letters are A, T, C, G, but write them out on your own chart. Adenine, thymine, guanine, and cytosine. A, T, C, G. So over here on this one, maybe one of the simplest ways to do it is just say sugar, phosphate, and one base, and just do those letters. Over here on your chart then, take it over here with an arrow, and generally on these, the phosphate is going to be shown here, the sugar is going to be shown here, and circle out this and just put any of those 4 letters. It really doesn't matter. I'm feeling T this morning. Now, as you work through this as we go down, go ahead and if you made this one large, which is fine, chart out a few others. Doesn't matter. Etc. So sugar, phosphate, and the base. The base is going to be in this open area. Now the DNA molecule does have another side, and at the very end of this we'll talk about putting that other side on, but for right now this part will work and it will make sense as you get a little bit further through working your chapter readings right now. Now on this one, nucleotide, a sugar, phosphate, and one base. Over here in the output column, I'm going to go ahead up here. What a sugar, phosphate, and base, or a nucleotide, does in terms of output is that it's the building block. So lots of us, when we're first learning something new, that we're a little bit nervous that maybe it's going to be complex or a little bit confusing, we actually do better if we have a visual image of something we're already familiar with. So think of a nucleotide as a Lego brick that comes in only 4 colors. Let's say red, blue, black, and white. That's just an idea. Or bricks. Are you more of a hands-on building sort of thing in reality? So bricks, clay bricks, you've got a whole pile of them. Some are loose, some are starting to build something. But if you have only one, it's a component. The same thing with one Lego. So over here on output, actually biochemically, one nucleotide does not have any output at this particular point in your discussion all by itself. It hasn't built up into something, right? That's why we started this going so long this way. So we need to add some more parts to this, right? All right. Okay, so next level down, I'm gonna just switch to a purple because I found it on my thing. So if you go down here, so I say if one nucleotide doesn't make any output, then it can't do anything to cause your system to run or any other organism systems to run, so it needs to come in groups. In this case, nucleotides come together for function in groups of 3. So I'm going to just say nucleotides times 3 make the next particular grouping. Now, lucky for you and me, when I was an intro level learner too, vocabulary for life science is often pretty clear and common. And in this case, 2 thumbs up, really, really clear and common. 3 nucleotides are called a DNA triplet. A triplet. So how would you chart that over here in your structural diagram? Just say, if you've got one of these and we've got 2 more here, I'm going to just say the next one could be C and a C, that it doesn't really matter. Then go ahead and bracket 3 of these, TCC, and that equals a DNA triplet. If we drop down over here, if we're moving this way, we need to leave a gap because there's a function that happens here that we want to label in, and then there'll be an output over here. So a DNA triplet. It has 3 parts to them. Every living thing that we have ever discovered on planet Earth is made up of sequences of DNA triplets. How many there are and how they express is how we know whether we're looking at one species of organism or another. So DNA triplet for here. So we're going to hold on for this section. Now, as you go through later on in the reading, and let me look and tell you what page it is for these, you get to one of the charts in the chapter that you're probably hoping we're going to ignore. On the other hand, incredibly important. So DNA triplet, and this part of functional information, you're going to see it on page 41 in your reading. It's table 3.1, and the big level says the genetic code. You drop down next one and it says amino acids. You drop down one more, right? One more level on your chart, and I hope you're looking at it right now as we talk about it. And it says DNA triplets next to something that's labeled RNA codons. So here's where triplet meets that term codon. So RNA, the other nucleic acid, it comes in and makes some changes or alterations. And again, you're going to see this as you read through the chapter. For us on our basic chart, though, we just want to show where that actually occurs so that you remember that they have something together. So RNA changes the DNA triplet into something that we term a codon. And codons have a biochemical output, a function. So if we drop down over here, so one of them doesn't do anything, but an RNA codon makes one amino acid. We've got that table there to take a look at. Okay, so I'm going to stop right here for a moment just to keep this a little bit short. Go ahead and take a look at that part of your reading and we'll come back to it in a couple minutes.